Hello, my name is Ravi Vij. I'm from My Cancer Haven. And today I have with me Dr. Ken Shain, who is co-director of the Pentecost Family Myeloma Research Center at one of the larger facilities focused on myeloma in the United States at Moffitt Cancer Center. He brings a very diverse perspective because not only is he a physician, he's also a researcher, and we will talk about some of the research that he's been doing in this area. And so today we're talking really about uh, personalized therapy in multiple myeloma. Where are we? Uh, and so welcome, Ken.
Well, thank you very much for the invitation, and thank you very much for letting me participate.
So, Ken, can you give us a little historical perspective on? We've made great progress in myeloma, and we are somewhat the envy of a lot of our colleagues in other disease states that we have over the last 20 plus years, maybe 25 now, got so many drugs. How did these come to be, and how are things maybe going to change in the future?
Yeah, Ravi, great question. I think one, I'll echo what you just said, is that I always tell my patients, no one wants myeloma, but today is the best time in the history of time to have myeloma because of all the options that we do have and the success we've had over the last decade, 2 decades, 3 decades. But when you think about what we've done is we've really come to a scenario where our therapies, we finally learned we need to use therapies that are based on targeting the disease, the plasma cell, the actual site and the source of the disease. Okay. And all of our drugs are used that way. So we've defined in drugs that work and kill plasma cell biology, but they still kind of are targeting everybody. They're not unique. They're not one patient, one drug. They're not personalized in that setting, but they've been incredibly successful over time. So we know that we have things that are called proteasome inhibitors, right? They target very specific activities that are highly active in these cancer cells, these myeloma cells. Immunomodulatory drugs are now called cell mods, where they're again targeting very specific plasma cell biology. And I think the next kind of big jump was looking at monoclonal antibodies to CD38, where again, targeting something on the surface of myeloma cells. These are all things that have been incredibly successful and helped our patients do a lot better, and thankfully have been very well tolerated for the vast majority of patients. But I think we're at that point where maybe we could really take all that information and try to better personalize therapy. So I can make every individual's patient decision based on more than just they have myeloma and they're newly diagnosed, more than they just have myeloma and their early relapse. We move to a more personalized setting. That's I think what we're kind of thinking about and how we can move our entire kind of management strategies forward.
So you said that we only now talking about getting into personalized therapies, and you mentioned that all these drugs have effects on the myeloma cell, but also probably on the microenvironment. So were these not the products of steady research and saying that we are going to understand the biology and then we're going to make a drug, or was it sometimes reverse engineering?
I think a lot of times it was reverse engineering. I mean, I always want to I always look at it this way. I spent a lot of my formative years growing up and getting my research was in how the microenvironment was making patients resistant, or how the microenvironment was making patient cells resistant to drugs. And I'll never forget, I was testing on a lab bench 4 drugs called IMID-1, IMID-2, IMID-3, IMID-4. Okay. They didn't work in my assay system very well at all. 3 of those IMIDs are fantastic: thalidomide, lenalidomide, and pomalidomide. So obviously work really well in patients. And when we were using them, we still didn't know how they worked. It wasn't until a decade plus later we actually learned how they work. We just knew they killed bad guys, maybe helped stimulate some good guys, i.e. T cells and NK cells, but we didn't know how they really worked. And it wasn't until later on that some really smart folks said, oh, there are actually these things that drag really important transcription factors to get destroyed so we can turn them off. And that's how they're killing bad guys. So I would say a lot of it's reverse engineering. Some is the right direction. Proteasome inhibitors kind of made sense, but a lot of it is that reverse engineering where they work really well. Let's figure out how they work later on. And I think that's one of the things that either way, we don't care because we're successful, right? And they work really well. But I think those are things you can look at different drugs, kind of take a different path. I think some of the CD38 antibodies made sense. We worked on it the correct direction. But I think what we've learned is, again, for the vast majority of people, these all help and have made great leaps forward by using different combinations in different settings. But one of the things I think we all need to strive for is, we recognize that maybe not all patients need all the drugs or they need a sequence of things. We have to figure out how do we do this on an individual basis or a small group or subclass basis.
So as far as personalized medicine goes, I know different people have different, definitions for personalized medicine. Some even say we got to personalize it to the patient's own health, their other health problems, what their social situation is. So you personalize in that regard. But what you're really talking about is personalizing it based on biology. Is that what you're
Yeah, it's a
getting at?
really good point to make. And so, yeah, I agree with you 100%. We always personalize therapy, right? We always make sure we're taking into account the person across in the room with us, right? How do we take care of them with the therapies we have to enjoy, to make sure they enjoy the highest quality of life with the highest efficacy? But yeah, when I say personalized medicine, I'm kind of taking one step deeper, which is can we identify aspects of the patient's disease or aspects of the patient's disease's microenvironment, right? The bone marrow, plus all the other critical things, right? Social is important, comorbidities are important, all those things are really important, but how can we maybe characterize patients into different groups that might benefit from different ways of treating them or different drugs at different times? And that's kind of what I think when I say personalized therapy, I really mean that level of defining management that we can make sure we understand what it is, not strictly speaking the stuff that's always important, which is the patient's right, the patient helps us make decisions, and all those factors go into it, but how do you define the biology of their cancer or their microenvironment, i.e. the bone marrow and their immune system?
So I guess to start to personalize, you first have to understand the biology. So can you give us a little primer on what we've learned about the genomics of myeloma? It's been, I think, already maybe 10 years since this first publication came out. And where are we
Yeah,
with
so
biology?
I think it's really we've come a long way in terms of understanding the molecular makeup of myeloma. I would argue, or at least just say out loud, that really we're not where we need to be yet. Okay, there's been a lot of enterprises around the world trying to figure out exactly what's going on. We've learned from myeloma is that unlike other kinds of cancers, there isn't one driver mutation that makes it myeloma. There is a kind of a smattering. And in fact, we just published a paper in uh, Journal of Clinical Oncology earlier this year with our colleagues over at Sylvester and a bunch of folks around the country and the world, actually, looking at 2,000 patients worth of whole genome sequencing. And we divided people into about 12 different subgroups. And the whole point was we could divide them up and we looked at their therapy using artificial intelligence kind of means and saying, hey, really, these patients might have benefited if they got XYZ, but they might not have needed transplant. They may not need these. These are things that we want to make sure we're moving towards, but it's not an easy process. And so, as we're talking about here, these early publications showing how heterogeneous or how different everybody is with myeloma makes molecular characterization difficult to target one thing. We know there's essentially 81 or plus driver mutations, but really the big ones are NRAS, KRAS, and they're not even always clonal. I mean, they're not always dominant, so they may not be the perfect scenario for a drug at a given time. So really, it's how can we group patients in terms of how they may respond? What does their DNA damage profile look like? These are all things we're working on and adding into overlying this genetic parts of the disease itself. And I think even beyond that, we probably need to know a little bit more about the biology. So we've talked a lot about genetics, et cetera. One of the things that we do all the time is we try to figure out what is the gene expression profiling going on that helps define a patient's biology and what might that be that makes them responsive to one drug versus another drug. So there are a lot of layers, but the goal is can we get the right layers and the right story to get people in the right cohorts to help this therapy versus that therapy, or maybe not a therapy. Maybe they can stop drugs. Those are all things we want to work on. And this is again in the context of new clinical findings, new clinical assays. All those things are critical to put in context.
So you mentioned that we have 80 plus mutations often in a single patient's myeloma, and then you mentioned that some of the successes have been disease states where we have one what we call driver mutation, like chronic myeloid leukemia, where we know that there's one mutation. But then we've had successes in diseases like acute myeloid leukemia. What makes myeloma different than acute myeloid leukemia that they have had successes? And we'll talk about some of the successes we've had, but not as many to show for.
Yeah, I think it's challenging question to ask, and I think it's really goes down to kind of the plasticity of what myeloma is, the heterogeneity within a patient itself, right? So there's multiple different clones that exist within a person at a time. So we call it a monoclonal disease, right? It's monoclonal protein, monoclonal this, but really it's several different clones, one at different sizes that are contributing to the disease at a given time point. And importantly, they're driven a little bit by evolution, right, to get to myeloma, but then they're also driven by the therapies you give it. So as you give therapy XYZ, certain clones will disappear, certain clones will start growing later on that were almost unmeasurable, if not unmeasurable at all at the beginning, become dominant clones because they're now resistant to therapy that they started with. And so you have these kind of clonal tides, as we're kind of pictured here, where this happens throughout the course of the disease, and these clones come and go. And that's one of the things that makes myeloma challenging. I think it's just part of the heterogeneity, the plasticity, the dynamics of our cancer that we focus on is it's not a simple cancer. It's not one big mush of one gene driving everything you can target, like you said, like CML, okay, or even some of the AMLs where you have certain mutations that you can drive on top of their induction therapies, is that we need therapies that can knock out a bunch of different clones and hopefully keep them away for a long time, but recognize we need to be able to have that chess match going on. What are we going to do next? Because it's going to drive some kind of resistance, and there'll be a new biology you have to target the next time. And that's why we have a hard time these kind of magic successes. But I would say that the drugs we do have now are successful because you have enough of them that target different biologies and different combinations. We can't anticipate these new biologies we have to overcome. And so that's where I think the success has come, although not as neat and tidy, so to speak, as maybe other cancers might be.
Sure. So I guess, these clonal tides are something that we can't at the moment follow clinically and tailor our therapies. But we are starting to have a few successes to show for. I understand that one of the things that may have had some utility has been this BRAF mutation. Can you tell us a little bit about what we know here and what other mutations in myeloma we may have had some success with, if any?
Yeah, it's a great question. Again, I think small success is a good answer, right? But it's a success that helps some patients, okay? And BRAF is a specific oncogene that we have, in lots of cancers. It's really enriched for melanoma, so skin cancers, again, not myeloma, melanoma. It's also seen in other kind of cancers that are solid tumors. And they developed drugs that target these BRAF, okay, called BRAF inhibitors, and specifically this mutation that called V600E. And then very nicely, there have been a couple reports talking about how if the clonal burden is high enough in a patient and they have a BRAF mutation, what happens in about 4 to 8% of patients with myeloma, okay, so a small fraction, right? We're talking about a small group of people, but does happen, and maybe be a little higher in incidence in those that have relapse disease and maybe disease that is no longer just sitting in the bone marrow itself. But the bottom line is that by giving these oral therapies, they will work for a period of time. They'll be able to knock down the myeloma that's driven by this BRAF. So I'd argue this is a really exciting kind of targeted therapy, okay? Precision medicine, so to speak. You have BRAF, use BRAF inhibitor, mom can go away for a while. And I've had anecdotes where patients have done very, very well when not much was left for them to do. And it's in fact one of the things that we do here is on an annual basis, we have a lot of analysis, mutational analysis done on our patients for research purposes, and we're allowed to screen, and we'll find a list of BRAF positive patients and make sure that we can use honest brokers to go back and let our physicians know, here's your patient that has this just in case you need this as an option going forward. It needs to be validated clinically, et cetera. But this is one of those things where I think really it's a place where when you don't have other options, it's a really exciting thing for patients to hear about if you carry this mutation. But again, it's specific. You have to carry this kind of mutation. And again, this MEK inhibitors kind of work together, have been shown to work as well against the same group of patients. FGFR receptor mutations are our targets for that as well. We might be looking at, there's other things that are along those lines. But again, mutation-wise, BRAF is still rare. The other ones are even more rare. I think really interesting will be as we develop more and more RAS inhibitors, mutant RAS inhibitors, how's that going to translate? Because they be maybe a little bit more dominant in our disease state. But again, those are things we have to figure out. I haven't seen a lot of any preclinical work in that scenario. So I think a really cool question. and a really cool space to demonstrate that it can work in myeloma. But again, myeloma figures ways around it in the end. It's not this all gone kind of disease state. It's just going to help us temporize and have our patients around longer.
So obviously, one of the successes or semi-successes, I guess, we've had because we still haven't had an FDA-approved therapy, but is this 1114 translocation. What can you tell us about what we know and how we can target this specific mutation? I mean, it's actually a translocation.
Yeah, so Ravi, as you well know, is that 11 translocation 1114 is driving the cyclin D1 oncogene to drive the myeloma cell growth, and it's about 20% of myeloma patients will have the 1114 translocation, and by itself isn't driving BCL2 expression, but really what we've learned is that it demonstrates an earlier phase of development in plasma cells. So these cells are still dependent on something called BCL2 for survival or not to dying. And venetoclax, as you kind of alluded to, is a BCL2 inhibitor, which if you inhibit this protein working, cells die. Okay. So a fantastic way of thinking about it. And again, as you said, I still can't figure out how we haven't gotten this drug FDA approved. Trial design, whatever it is, bad luck, I don't know. But this is a therapy we know works extremely well in 1114 carrying patients. Um, we have lots of early phase data demonstrating that it does so with Dex alone in combination with different components, monoclonal antibodies to CD38, et cetera. Um, and it's so successful that, if you're a patient of mine and you have 1114, as soon as you are relapsing on X, Y, or Z, I'm trying to figure out how to get venetoclax for you in combination with, again, daratumumab or carfilzomib, things like that, because I think it's that important to get it to patients early and get it to patients as soon as they can. But it is an amazing story of a successful drug that in the myeloma space, we haven't been able to get approved because we've designed trials that haven't been positive, quote unquote, even though they still have very good data. So it's a really challenging thing. Thankfully, it's approved in lymphoma. Thankfully, it's approved in AML, so we are able to get it for our patients off label. But again, I think it's a great example of precision medicine in myeloma and highly successful therapy in myeloma. But yeah, I have to tell you that I still struggle with how we haven't figured out how to design the right phase 3 study to make sure the FDA is happy about it. So I don't know if you have any comments on it either, but I think it's a little bit of a frustration on my end.
Oh, I totally agree. And I think that we still have faith in the drug and we get it, fortunately, for a lot of our patients through insurance or through the manufacturer. There are other BCL2 inhibitors in development. Hopefully, those will be more successful and we'll maybe have an FDA-approved treatment one day. But I guess venetoclax still, that time is still worth pursuing for these patients. Wouldn't you agree?
Oh, 100%. Like I said, in my practice personally, if you carry this mutation, I'm already thinking about how am I going to integrate this into your therapy as quickly as possible. And again, I think biology of myeloma tells us that earlier use is better than late use. I think you lose that BCL2 dependence as you go on. So I try to get it in, second, third line, fourth line as fast as I can in that space. Again, in combination. I think it's just that good a drug and that well tolerated a therapy too. I mean, it's most patients have very few issues with it. So again, a great example of I have a biomarker, translocation 1114. You could argue high BCL2 ratios, et cetera. But again, simply 1114, and it works really, really well. So I love it. Let's just put it that way.
So now, obviously, you mentioned about clinical trials and our inability to sometimes get drugs approved. But I guess in the era of personalized medicine, the whole concept of how we do clinical trials may also have to change. So can you tell us about how you think in the new era where we're looking at diseases like myeloma, where there are so many different mutations and not a few select group of mutations, how we can approach this problem?
Yeah, I think, as you have on the slide, you kind of have how we do things now, which is we bunch everybody together as myeloma in a certain disease state, and we test drug X, Y, Z or new drug plus X, Y, Z, and kind of move it through the phases until we get randomized control studies and treating myeloma as a group, right? In that setting, like relapse disease versus newly diagnosed. But I think as we've kind of alluded to along the way is that We know myeloma isn't that simple. We know there are specific kinds of mutations that hold through in groups of patients. So maybe we should be thinking about things like umbrella trials or basket trials, which are terms just to describe patients that may not be myeloma, but they have this mutational pattern. So we get drugs that target that mutational pattern, even outside the context of myeloma, multiple drugs, sorry, a drug across multiple cancer states based on the biology, based on the molecular characterization of the disease. Or you have like my drug we have in myeloma, where we use myeloma therapies plus other drugs that target these mutations, okay, along with standard of care myeloma therapies to try to drive these specific individuals in a better place and a better way of doing things. So I think we really need to think about how we're doing trials today, but how we can better allocate trials and get people into the right studies based on their molecular, again, personalized kind of ways of looking at trial design.
So umbrella trials obviously break the disease into multiple subsets and try different drugs for the different subsets. Basket trials actually do away with the concept of disease itself, just have a mutation and put all the other disease states with that mutation into one trial. So
Absolutely.
obviously these are new concepts in trial design that continue to be explored. But I also know that you have at Moffitt for a long time been pursuing personalized medicine, and you have your own platform. So can you tell us a little bit about how you're approaching this dilemma?
Yeah, thank you so much. And I'd love to talk about it for the next 45 minutes or so. Just kidding. So, we've done, as we said, how can we take samples and how can we do something in real time? So when you think about myeloma, it's a disease where we talked about the molecular aspects of the disease. We need to get sequencing, we need to get mutational analysis. But can we do something that might be even a little closer to what's going on? So with a partner here at Moffitt, Ariosto Silva and I, we developed a study and a platform where we can actually, when patients get bone marrow biopsies done, we can then isolate their myeloma cells, and we've created an entire system to then test their cells, their myeloma cells, against a panel of up to 31, in fact, up to 127 drugs or drug combinations simultaneously. And so essentially that patient's undergoing, their cells are undergoing 31 different clinical trials simultaneously. And the goal is, can we test how their cells will respond to those individual drugs or combinations? So we have a bunch of standard of care ones plus experimental ones. And from there, how can we model that to then predict the clinical outcome so that we can then provide physicians, this is what your patient will respond to, RVD. Yep, they're going to get a partial response. KRD, they're going to get a complete response. So how can you better derive therapy? And so we built a system to be able to give us this answer in about 7 days. So it's one of those things where we have developed this system. We've been doing it for a number of years now. And so that's one level of it. And again, we've learned that some drugs we can do really well at, some drugs maybe we're not as perfect at, let's put it that way, because they don't have as much activity as we need to, or we're just modeling it a little incorrectly. So we're still working on how we can tweak that a bit. And then we've done we call it EMMA, okay? Ex vivo myeloma mathematical advisor. But now we have EMMA 2.0, where all those same samples have RNA sequencing and whole exome sequencing done. So all the things we just talked about, mutations, gene expression, we can now see what drugs in that patient worked or didn't, and how does that correspond to a biology. So now we can group patients into larger groups, and so how do we figure out what's going on? We call it EMMA 2.0. So now we find the genetic and or the biological states that make you sensitive to this drug or not. Now our next hope is, can we apply that to individuals and hopefully, again, find the right drug for the right group of patients, maybe not the patients with this kind of biology. And what's really cool is we're trying to get something published right now where we've learned because of this is that certain drugs, if you give them the right sequence, will give you better outcomes. So drug X predicts and creates a biology that makes you sensitive to drug Y. And so that would be great if we could actually figure out what sequence to use drugs in. We'd increase that control of disease across time, and therefore our patients would live longer and better, but just by giving the right sequence of the drugs we already have. So I think those are things we're all working on, and I think it's a level maybe a different. I'm not saying better or worse than the molecular way of doing it, but I think it's going to help us really figure out better ways to allocate therapies to patients. Again, that's our goal.
So that's certainly wonderful. It seems like you're not just saying we're going to try to go after the mutations, which are, as you said, so few recurrent mutations that you can really develop drugs with, but you are trying to just take the cells and study their sensitivity or predict their potential sensitivity to uh, based on the whole genomic profile, so as to say, and say that, these drugs we use anyway, or these drugs are used in other disease states, and based on the findings of the whole analysis that you do using your computer algorithms, that this drug is likely to work best. That it, Velcade will work for this patient probably, but may not work for the other one.
Yep, 100% correct. And that's our kind of goal. And even the combination, right? So 3 drugs, 4 drugs, you might not need all 4 drugs, you just need 2. So yeah, it's exactly what we're looking for is how can we improve the utilization of the drugs we have, and how can we integrate drugs that we don't use because they're attacking the biology, again, not just the mutation, but the biology of the patient. Because again, the biology drives all this. And mutations, I think, are just a part of the story. And I think that's how we've kind of done it. Now, it may end up being integrating everything in the end. We'll have to do it. But again, this is our way of looking at it at our level.
Yeah, so there's a sort of dichotomy in cancer therapeutics, where some people have, at least in the past, been focused on the molecular characteristics, going after the molecular characteristics, and then people say, well, no, we can just treat it all with immunotherapy, because immunotherapy is an equalizer and doesn't depend on the mutations per se. But that's not also true, because we know, like, checkpoint inhibitors, which are used in solid tumors, it appears are more likely to produce responses in patients who have a lot of mutations in their cancer. So I guess it's not like, I guess, two different things. It's like they can come together.
100%. I think in the end they're going to need to. I mean, one of the things that we need to think about is how we marry these two aspects. So there's definitely going to be a genetic component to it, but the genetics are kind of a fingerprint at a point in time, and they will change a little bit as we talk about those clonal tides change. But in reality, I think a lot of the mutations that occur in myeloma are all driving a very similar biology. So the biology may be what you're targeting, not necessarily the mutation itself, but they may be critical to how you get there. And so I think these are really important things. And immunotherapies, absolutely, they're revolutionizing how we take care of disease today, but it doesn't mean they don't need to be personalized as well. I mean, our early results are saying that there are some things that might make you resistant to immunotherapies, whether it be the target being there or not. I mean, those are things that have been published already, right? If we have loss of antigen or we don't have the target for immunotherapy to hit, it's not going to work. So that's one level of personalized therapy. But there may also be an even more global kind of change, that is, these patients don't look as much like myeloma as they did before. It might not work as well because they're not as plasma cells want them to. These are all things we have to figure out. And that's where genetics and I think biology, whether it be proteomics, transcriptomics, epigenetics, whatever it is, they have to kind of come together. And then I think our drug assay kind of gives you the final story. This is what they're going to be sensitive to or not, because of all those things that go together to lead to that phenotype, which is drug sensitivity. So I think these are all things we need to really continue to work on. And I think with AI and newer technologies, we're really learning how we can put this data together to hopefully make this a reality, right? Like you and I talking right now, it's something we're working on. It's not something we know is going to work 100% of the time, and we hope we get there, right? We anticipate getting there. But I think these are critical steps we have to do to explain the different parts, to put them back together to give us the best chance of helping our patients, which is what we're all here to do.
That's amazing. And obviously, you're at the forefront of trying to do so, to personalize therapy for patients based on their biology as a whole, rather than just one mutation, or just blindly throwing drugs at patients and hoping they work. I think your research will be something that we would love to follow in the years to come, and wishing you all the success. I hope I can have you back in the future to talk more about your own research and other topics in myeloma as we make advances.
Robbie, no, thank you very much. This has been an absolute pleasure. Thank you for the invitation. I would be happy to help in any way I can. And now it's recorded, so I can't back out. So we're all set.
Thanks a lot, Ken.
All right. You take care. It's great seeing you.
Bye bye.